Thus far, we focused on describing different ways of extracting information about the function of a genetic device in a cell. There are additional considerations such as the choice of controls and choice of experimental conditions that must be included in the test phase. We'll describe 4 specific areas of information one needs to extract from an assay. In differential expression, you are comparing a strain's function under 2 or more environmental conditions. In a transfer function, you are continuously varying some environmental parameter and trying to produce a function relating the output behavior to this input. Dynamics involves following the system over time with your assay. Finally, population heterogeneity involves performing your assay on individual members of the cell population. Differential expression involves performing an assay under different conditions. In this example, we are trying to characterize the response behavior of a magnesium-responsive promoter in LB medium versus PBS medium. The genetic circuit places the RFP gene under the control of the promoter we wish to test, or a control constitutive promoter. Red fluorescence and OD are measured fluorometrically after growth in different media. The signature of a differential expression experiment is the non-continuous nature of the input variable. In this study, those different inputs are specific media compositions. In many industrial-scale fermentation processes, these inputs might be different chemical feedstocks. The conditions employed are specific things, and it is not relevant to ask about a condition halfway in between. A transfer function experiment involves scanning through a continuous environmental variable. In the previous example of the magnesium-responsive promoter, we would grow the cells in LB medium with many different concentrations of magnesium and then plot red fluorescence over time. Here we have the same thing, but a different piece of regulation and a different environmental input. It's the PBAD promoter now driving GFP instead of RFP, and we are scanning through concentrations of arabinose instead of magnesium. It is usually possible to derive kinetic models of regulatory systems like this and predict the shape of these functions. And often folks will make these models and then fit the parameters to fit the model. This then allows interpolation to intermediate values for the input. In this example, we are showing the response of an AND gate of 2 different inputs, arabinose and salicylate. Thus, the inputs of a transfer function can have more than one axis. Finally, we have an example of a continuous range of inputs used to construct a transfer function. The cells here contain the light sensor circuit from the Voigt lab driving LAC-Z. They put a layer of the bacteria on the surface of a petri dish and then shine light on it through a continuous mask. When the light level is right to turn the system on, LAC-Z gets produced and sGal is turned into a black pigment. They then use image processing to quantify the darkness over the gradient to plot out the transfer function. I'll show you these 2 clips after this presentation, but these are 2 examples of following the dynamics of single cells containing a genetic circuit over time. These oscillating genetic circuits require monitoring the behavior of individual cells over time. Currently, the only way to do this is fluorescence microscopy, and thus such assays only work for fluorescence readouts. It would be extremely challenging to, for example, monitor single-cell dynamics of a cell biosynthesizing a biofuel. Another measured property of a strain is population heterogeneity. Here, one cell in the population does something different than other cells. Sometimes this is an intentional property. For example, this variant of the toggle switch from the Peking iGEM team in 2007 is designed to cause population heterogeneity. Thus, they need to look at individual cells in the population and measure 2 different channels of fluorescence to see what they want to see. However, often population heterogeneity is an unwanted and challenging to measure property of a strain. For example, you might have a biosynthetic pathway in a cell in which only 10% of the population is producing the genes needed for biosynthesis. With bulk measurements, it is impossible to know that such heterogeneity exists in the system, but it is challenging to decipher this quality of your design directly. If the heterogeneity is due to genetic mutation, this can be pulled out from single-cell sequencing. There are efforts to develop other single-cell assays in the works, but as of 2014, sequencing and fluorescence measurements are the only off-the-shelf way to ask questions about population heterogeneity.